In drug safety assessment, accelerating the evaluation of molecular toxicity is crucial for efficient drug discovery. To address this challenge, researchers have applied bidirectional long short-term memory (BiLSTM) networks to analyze molecules represented in Simplified Molecular Input Line Entry System (SMILES) notation. By treating SMILES strings as sequences, the BiLSTM model can capture sequential patterns that inform toxicity predictions. In a comparative study, the BiLSTM approach outperformed previous methods, including Trimnet and Pre-training Graph neural networks (GNN), on the ClinTox dataset, achieving a high ROC accuracy of 0.96. Furthermore, the BiLSTM model demonstrated superior performance on the FreeSolv dataset, yielding a low root mean squared error (RMSE) value of 1.22 in solubility prediction. This work underscores the potential of BiLSTM in expediting drug discovery by accurately predicting molecular properties. By leveraging the sequential nature of SMILES notation, BiLSTM networks can accelerate toxicity assessments, ultimately streamlining the drug development process. The proposed model utilizes Paracelsus' principle to predict toxicity at doses relevant to patient use, distinguishing between toxic and non-toxic effects based on dosage levels for each drug. It uses SMILES as input and employs a tokenizer object from TensorFlow Keras for character-level tokenization. The model is designed to overcome the limitations of previous methods, particularly in terms of the root mean square error, RMSE, which is greater than 2. It leverages recent advancements in cheminformatics, utilizing deep learning techniques such as graph neural networks, GNN, for molecular property prediction. The model is trained on datasets like ClinTox, TOX21, and synthetic data, providing a comprehensive framework for assessing toxicity and predicting molecular properties. In this study, a novel approach is employed to predict the toxicity of compounds by leveraging the strengths of bidirectional long short-term memory (BiLSTM) networks and graph convolutional layers. The methodology involves processing input sequences in both forward and backward directions using BiLSTM, thereby enhancing the ability to capture correlations between sequences. The encoded inputs are then fed into the BiLSTM layer, which processes them bidirectionally to extract comprehensive feature representations. The output from the BiLSTM layer is subsequently passed through a dense network equipped with a sigmoid function to predict toxicity. This model is designed to elucidate the relationship between molecular structure and toxicity, providing valuable insights into the underlying mechanisms. Notably, graph-based methods have proven instrumental in predicting molecular properties, and the graph convolutional layer plays a pivotal role in combining information from nearby nodes and edges. By integrating these two powerful techniques, the proposed model is capable of capturing complex patterns and relationships in molecular structures, ultimately enabling the accurate prediction of toxicity. The paper discusses the representation of molecular structures using Simplified Molecular Input Line Entry System (SMILES), and its application in a neural network for property prediction. SMILES is a notation system that encodes molecular structures into a unidimensional string, with symbols representing different atoms and bonds. This string is then converted into a binary array and inputted into the neural network along with the target label for analysis and prediction. The paper also introduces long short-term memory (LSTM) networks, which are effective for tasks involving sequential data due to their ability to retain and selectively utilize information over time. LSTMs have feedback connections that allow them to store the results of the current input for use in the near future when making other predictions. They are particularly applicable for tasks like text recognition, speech recognition, language translation, and sentiment analysis. The LSTM model uses a gate mechanism, which includes three main gates: input, forget, and output gates, as well as a cell state that acts as a memory. Each gate has specific functions, such as deciding which information from the current input should be stored in the cell state, input gate; which information from the previous cell state should be forgotten or discarded, forget gate; and what information from the current cell state should be output to the next layer in the network, output gate. The equations for the LSTM gates are provided, including the forget gate, input gate, output gate, and candidate gate output at time step t. The final states are represented as the product of the forget gate and the previous cell state plus the product of the input gate and the candidate gate output, and the hidden state is the product of the output gate and the hyperbolic tangent of the cell state. The proposed model architecture consists of 3 layers: an embedding layer, an LSTM layer, and a dense output layer. The embedding layer converts each SMILES sequence into a dense vector representation, suitable for processing by the LSTM layer. This LSTM layer processes the embedded SMILES sequences, capturing dependencies and patterns within the data over time. The final dense layer performs the final classification based on the LSTM's output, predicting the toxicity, which is a binary label. The LSTM layer is further explained, where the forget gate, input gate, and output gate are introduced. The forget gate determines what information to discard from the previous cell state. The input gate determines what new information to add to the cell state, and the output gate determines the final output of the LSTM cell. The cell state and hidden state are updated based on the gate's outputs, as illustrated in Figure 2. The update equations for the cell state and hidden state are provided, where quart represents the cell state at time t, and ht is the final output of the LSTM cell. The gate's outputs are calculated using the sigmoid and tanh functions, which introduce nonlinearity into the model. The architecture is applied to 2 datasets: the toxicity dataset, where the model predicts the toxicity of a molecule, and the FreeSolv dataset, where the model predicts the regressive values of molecular solubility in moles per liter. The same procedure is followed for both datasets, with the LSTM layer capturing dependencies and patterns within the data over time, and the final dense layer performing the final classification. The dropout layer is also introduced, which randomly sets a fraction of the output units of the LSTM layer to 0 during training, preventing overfitting. The output layer uses a sigmoid activation function to produce the final classification output. The Bidirectional-LSTM architecture is designed to predict molecular properties from SMILES strings. It begins with an input layer where these strings are fed into the network. An embedding layer then converts each character into a dense vector representation. The BiLSTM layer processes the input sequence in both forward and backward directions, capturing past and future dependencies in the SMILES sequences. The output layer combines the hidden states from both LSTM networks to predict molecular properties. The gates of the LSTM are sigmoid activated, ensuring values range from 0 to 1. This architecture is illustrated in Figure 3, showcasing the proposed model's structure using BiLSTM. The BiLSTM model's architecture, illustrated in Figure 4, facilitates the capture of contextual information from both past and future sequences, thereby enhancing solubility prediction performance. During training, gradient descent optimization algorithms, such as ATOM, are employed to minimize the mean squared error (MSE) loss function, a common choice for regression tasks. Comparative analysis reveals the superiority of the sequence-based approach, with BiLSTM models trained on SMILES data outperforming traditional methods in terms of prediction accuracy and efficiency. Notably, the model achieves exceptional performance on the ClinTox dataset, yielding an ROC accuracy of 0.96 on the FDA approval task and an average ROC accuracy of 0.96 plus or minus 0.01 across both tasks. This surpasses the previous highest model by 2%, demonstrating the efficacy of the BiLSTM approach in solubility prediction tasks. The comparative analysis of the proposed model's performance in predicting molecular solubility and toxicity is presented. Figure 5 illustrates the ROC accuracy comparison of models on ClinTox and Tox21 datasets, where the proposed model achieved a ROC accuracy of 0.81, surpassing graph-based methods like pre-training and relational pooling. Notably, BiLSTM outperformed GAN models in terms of ROC accuracy. When performed on synthetic data, the model achieved an ROC accuracy of 0.80, capturing complex relationships between molecular structures and solubility. Furthermore, the proposed model outperforms the previous best model GLAM with a lower RMSE value of 1.2 compared to GLAM's 1.31, indicating better predictive performance. Figure 3 provides a comparison of the BiLSTM model with previous regression models, highlighting its significant advancements in predictive performance. The proposed model, a BiLSTM network, has been optimized through experiments with various parameters to enhance its performance on the training data. Notably, the model achieved better ROC accuracy with the following hyperparameters: dropout rate of 0.3, learning rate of 0.1, and 32 units. The research demonstrates the potential of sequence-based methods in advancing computational drug discovery techniques, particularly in predicting solubility and toxicity. By leveraging SMILE's information, these models provide a more accurate and streamlined approach compared to traditional methods. The proposed model presents a computational approach for predicting drug toxicity based on SMILE's representations, aiming to accelerate the drug discovery process. Deep learning methods, specifically BiLSTM, have been explored to enhance the accuracy of toxicity prediction. The strong performance of BiLSTM is evident, especially when measured using the ROC accuracy metric on the ClinTox dataset. The use of LSTM models, typically employed in NLP tasks, for toxicity and solubility prediction on SMILE's representations is a novel aspect of this research. The sequence-based approach can be further enhanced by utilizing models like GPTs and LLMs, and by collecting and training on a larger amount of data. The research page discusses various contributions to the field of drug-target interaction prediction, molecular solubility prediction, and molecular property prediction. It highlights the SPVEC method, which uses a Word2Vec-inspired feature representation, and the SOL predictor model for solubility prediction. The graph networks framework for molecules and crystals and graph neural networks for molecular solubility prediction are also mentioned. Additionally, adaptive graph learning methods for automated molecular interactions and properties predictions are discussed, as well as the UniMol framework for universal molecular representation learning.